Assalamu alaikum. Hope all of you are fine. We're going to start with the development of the nervous system, and in this lecture we'll discuss the early development of the neural tube, the formation of the spinal cord, the gray and the white matter formation from the neural tube, and what is the role of the migration of the neurons during the neural development, and what is the contribution of the neural crest cells in the formation of the peripheral nervous system and central nervous system, the positional changes of the spinal cord, and then the developmental basis of the certain congenital anomalies like the spina bifida. As you know, the nervous system consists of the central nervous system, which consists of the brain and the spinal cord, and they are protected by the cranium and the vertebral column. And the peripheral nervous system, which is the outside the central nervous system and consists of the 12 pairs of the cranial nerves and 31 pairs of the spinal nerves, which connects the central nervous system to the peripheral organs. And the autonomic nervous system, which consists of the sympathetic and the parasympathetic parts and their associated ganglia. Now, the first indication of the development of the nervous system takes place at the stage of the 22 to 23 embryo in the region of the 4 to 6 pairs of the somites by the formation of the thickened ectoderm under the inductive influence of the notochord and the paraxial mesoderm at a trilaminar stage of the embryo. So this thickened ectoderm above the notochord forms a plate-like structure, and this is called the neural plate or the neuroectoderm. And this neuroectoderm is a thickened plate. It is visible on the posterior aspect of the embryo, which is at this stage is a slipper-like or a disc-like stage of the embryo. So the formation of the brain and the spinal cord is called the process of the neurulation, and this process of the neurulation takes place at the level of the fourth pair of the somites. This formation, the neural plate thickens and it deepens and it forms the neural groove. And so the cranial to the fourth pair of the somites, the two-third portion will form the future brain, and the caudal one-third portion of this neural tube, it will form the future spinal cord. So in the middle of the somites, the edges of the neural plate, they start folding in each other over each other, and they form the neural folds, so that they closes and this closure occurs in a craniocaudal direction, and the two pores they remain open, which are called the anterior neuropore and the posterior neuropore. Now, this anterior neuropore and the posterior neuropore, through these pores, this area, it forms the vertebral canal, and this communicates freely with the amniotic cavity. Now, this is another view you can see here, that this is the whole of the neural tube formation, and these thickened portions, they are the somites. So at the level of the fourth pair of the somites, you can see that the cranial portion, it develops into the brain, and the caudal portion, it develops into the spinal cord. Now, this is the thickened plate. First is the formation of the neural plate, and then the neural groove formation takes place in a trilaminar embryo. And this neural plate is formed from the thickened ectoderm between the primitive node and the buccopharyngeal membrane. And then this is the closure of the neural folds takes place to form the neural tube in a craniocaudal direction. And this anterior neuropore closes at the day 25th, whereas the posterior neuropore closes at the day 27th. And this diagram showing you all the events of the process of the neurulation. You can see here that under the inductive influence of the notochord, the neuroectoderm, it thickens and deepens to form the neural groove and the neural plate. This neural groove, they meets in the midline to form the neural tube, and the edges they are raised to form the neural folds, and these edges they are detached and they are called the neural crest cells. These neural crest cells they will migrate and they form the different structures. Now the development of the spinal cord. The spinal cord develops from the neural tube, but it is caudal to the fourth pair of the. This diagram showing you the thickened wall of the neural tube. This thickened wall of the neural tube is made up of the pseudostratified ciliated epithelium. The pseudostratified ciliated epithelium showing you the lot of the mitotic activity, and it gave rise to the different types of the cells of the neuroepithelium. The central canal of this neural tube, it will first it is widened, and then because of the thickness of the wall of the tube to form the neuroepithelium, it becomes narrow, and then later on in the spinal cord it just. Now, this layer of the neural tube, which is the layers of the cell which are close to the central canal, this layer is called the ependymal layer or the ventricular layer. Now, this ventricular layer or the ependymal layer of the cells, it gives rise to the all the types of the cells, and it forms the different layers. So first the ependymal cell layer, it forms the neuroblast cells, and these neuroblast cells they lodge in a central a layer which is called the mental layer. Now this neuroblast cells they will give rise to the neurons once they form the processes. So they start proliferating and they form the multiple neuroblast cells. Now soon a layer appears at the other end which is called the marginal layer or the marginal zone, which forms the white matter of the spinal cord, as it will have all the processes or the nerve fibers of the neurons. So the neuroepithelium, it differentiates into the different layers of the cells, and these cells in the ependymal zone or the ventricular zone, they are responsible for giving rise to all the cells. They will also give rise to the microglia cells, which includes the astrocytes and the oligodendrocytes. We'll discuss later on. Now, once these neuroblast cells they are formed and it ceases to form the neuroblast cells, then these ependymal layers cells it will forms the ependyma or the ependymal layer around, which surrounds the central canal of the spinal cord. Now, these cells they are the tall columnar cells. They are ciliated, and they will help in the circulation of the CSF. Now later on, this mental layer, which is actually the gray matter of the spinal cord, as it includes only the neuronal cell bodies or the neuroblast cells. So this mental layer, it will starts the proliferating and differentiating. So there are the different levels of the differentiation. So this lateral wall, it grows rapidly and it thickens, whereas there is the thin roof plate and the thin floor plate. So because of this differentiation of the growth, there is a groove which is formed. It is called the sulcus limitans at both the edges. Now this mental layer is dorsally it differentiates into a alar plate and ventrally into a basal plate. Now, this cells of the neuronal cell bodies in the basal plate, they will give rise to the motor neurons in the basal plate, and this horn is called the motor horn. So the all the neuronal cell bodies they are grouped into the here into the motor neuron, which are give rise to the fibers, which are the efferent fibers. Whereas the alar plate, they have the neuronal cell bodies, which are the afferent neurons, and they are the sensory neurons. So this is called the posterior sensory horn of the spinal cord. So you can see here now this the central canal, it will be reduced to a smaller central canal and the sulcus limitans which is surrounding it. So it will be shrink because of the growth of this gray matter. Now, this surrounding area around the gray matter is the white matter, which is formed by the marginal zone or the marginal layer of the neuroepithelium. Then you can see the ventral fissure and the posterior median sulcus of the spinal cord also. So this is again showing you all the events in the development of the spinal cord. This is the neuroepithelium, and these are the neural crest cells which will give rise to the different structures. And this is the ventral horn or the motor horn of the spinal cord, which is formed from the basal plate, and it will give rise to the motor horn, the neurons or the neuronal fibers, which are the efferents, and then this alar plate, which forms the posterior sensory horn of the spinal cord, and it has the sensory neurons, and they will give rise to the afferent fibers. So these afferent fibers, they will afferent and efferent, they will meet in the intervertebral foramina to form the trunk of the spinal nerve. And this is the dorsal root ganglia, which is formed from the neural crest cells. Now, the histogenesis of the cells of the central nervous system. So, as we have discussed that the cells in the ependymal zone or the ventricular zone, they will give rise to all the types of the cells. They show the highly mitotic activity, and they will also give rise to the glioblast cells or the macroglia cells. So these glioblast cells, they are of two types, the astroblast and the oligodendroblast, which will forms the oligodendrocytes, and the astrocytes, which are of two types, the protoplasmic astrocytes and the fibrous astrocytes. So these are basically the neuroglial cells or the supporting tissue of the nervous system. So they are also derived from this neuroepithelium from the ependymal layer. And as we have discussed that the neuroblasts, they are also formed in the mental layer of the neuroepithelium from the ependymal zone. So these neuroblast cells, they are the rounded in structure, and they are apolar. Then they differentiates into give the processes, and they form the bipolar or the unipolar or the multipolar neurons. And this is the neuronal cell body, and these are the axons, which will be lying in the marginal zone of the neuroepithelium, which forms the white matter. Now, another type of the cells, the ependymal cells, as these from the ependymal zone, which just lining the central canal, they are the tall columnar cells, ciliated tall columnar cells, and they will be they have the choroid plexus, and they will help in the formation and the circulation of the CSF. Then the other type of the cells, they are the microglia cells. These microglia cells, they are formed from the mesenchymal cells. They are not formed from the ependymal cells of the ependymal zone. So they are from the mesenchymal cells. So this layer is the mesenchymal layer, and these are the microglia cells. They are derived from the mesenchyme. Now, this is a diagram showing you the transfer section of the embryo at the stage 16. Now, you can identify all the structures which we have discussed. This is showing you the roof plate and the floor plate, and this is the central canal, and this is the sulcus limitans. Now, you can identify the alar plate and the basal plate also here, and this is the dorsal roots of the spinal nerve, and you can identify this dorsal root ganglion, which consists of the unipolar pseudo-unipolar neurons. And this is the mesenchyme, which will later on forms the vertebral body. Now, the derivatives of the neural crest cells. So, as we have discussed, the neural crest cells will be detached from here, and they will migrate, and they will give rise to the different structures. So, the neural crest cells, they will give rise to the melanocytes. They will form the parts of the peripheral nervous system. as the dorsal root ganglia, and they will form these ganglia of the sympathetic trunk. They form the celiac ganglion, the renal ganglion, part of the suprarenal medulla. So all these structures, they are formed from the neural crest cells. Now you can see here that the neural crest cells, how they differentiate into the unipolar neurons, bipolar neuroblasts. So the first they are rounded structure, then they give the processes to form the bipolar or a unipolar afferent neurons. Now, the process of the myelination of the nerve fibers. So the process of the myelination of the nerve fibers, it occurs in the late fetal period and continue into the postnatal period. So first year of the postnatal period. So in the peripheral nervous system, the axons or the nerve fibers, they are myelinated by the Schwann cells, whereas in the central nervous system, the myelination of the axons takes place by the oligodendrocytes. So first the axon, it indents the cell, and this indentation is deepened and it forms the mesaxon. And this cell, it binds rounds or wraps around the axon, and this wrapping continues so that the cell rotates itself around the axon, and the cytoplasm is squeezed between these membranes. So the cytoplasm, it remains between these membranes, and the cell rotates itself around the axons, and so that again later on this cytoplasm is squeezed and this myelinated layer is formed around the axon. Similarly, in the central nervous system, these oligodendrocytes, they have the numerous processes, and each process, it wraps itself around the axon. So the wrapping continues, and cytoplasm is squeezed between these the foldings, and later on this myelinated layers, they are formed around the axon, and then it is detached from this process. Now, the positional changes of the spinal cord. So, during the embryonic life, the spinal cord, it occupies the whole length of the vertebral column. And later on, the dura mater and the vertebral bodies or the vertebral column, it grows at a faster rate than the spinal cord. So, at the stage of the 24-week-old fetus, the spinal cord, it lies at the level of the S1. Whereas in the neonates or the newborns, the spinal cord, it shifted to a higher level at the level of the L3. And in the adults, it reaches at the level of the disc between the L1 and L2. Now, the congenital anomalies. There are various congenital anomalies associated with the development of the spinal cord, but they are called the neural tube defects. So, because they are due to the closure of the non-closure of the anterior or the posterior neuropores. So if there is a non-closure of the anterior neuropore, it will result into the encephaly, whereas the posterior neuropore non-closure will result into the neural tube defect, which is called the spina bifida. So the spina bifida is a general term for the neural tube defects in the spinal region, and it consists of a splitting of the vertebral arches and may or may not involve the neuronal tissue. So there are two types of the spina bifida, that is the spina bifida occulta and the spina bifida cystica. Now, in the spina bifida occulta, there is a defect in the vertebral arches, and usually does not involve the underlying neuronal tissue, and it is visible in the region of the lumbosacral, that is L4 to S1 level, and it is marked by a patch of the hairy skin over the affected area. So this is a less severe condition, The spina bifida cystica is a severe condition of the or a severe congenital defect of the neural tube defect in which the neural tissue is also herniated and form a cyst-like structure. So it is protruded out, and sometimes the meninges they are protruded out, and sometimes the whole of the spinal cord is also protruded out. So this is called the this is called the meningocele, and when the spine neuronal tissue or the spinal cord is also protruded out along with the meninges, it is called the meningomyelocele. Then another form is the where the neuronal tissue is absent. So there is no neuronal tissue is formed, and this is called the this is a sphere form of the neural tube defect. So this is all about, in this lecture, we have discussed the development of the nervous system, the primordial of the development of the nervous system by the process of the neurulation and the development of the spinal cord. So this is the first lecture, and we'll continue with the development of the nervous system in the next lectures. Thank you.